let's look at our schedule for a minute. See where we're supposed to be. So this is 7th. We're on chapter 3. Exam next week. Let's plan on doing that on Friday. So that our first exam covering chapters 1 through 3 will be next Friday in class. Multiple choice, maybe some matching. I think 50 questions. Questions similar to the ones on the sets of sample questions on Moodle. And reminder again, on Moodle, under this week, there's this homework assignment that's due Monday. To turn in on paper in class, so please have it done when you get here. Had a week to work on it, so hate to see students show up and then working on it during class when it should have been done. In plenty of time. Okay. So chapter 3. Where were we on here? Anyone remember where we left off?
We were at like cell theory. So we left off there at cell theory.
Cell theory? Okay, thanks. Oh, yeah, we just barely got started on this chapter, didn't we? Yeah. Okay. Oops. Okay. Yeah, okay, the so-called cell theory, every living organism is made up of cells. Four parts that almost every cell has. There are exceptions to even this, but every cell is covered by a plasma membrane, or sometimes called the cell membrane. It's just a thin layer of flexible material that contains the cell. The inside of the cell, what is surrounded by the plasma membrane, is called the cytoplasm, and some books include the nucleus as part of the cytoplasm. Other books do not. So I forget what our book, what the book even says that we're using now, but but every cell has DNA or chromosomes, and an exception to that is human red blood cells don't have chromosomes, and ribosomes. These are the little organelles in cells where proteins are made. So every cell has to be able to make proteins. And we already have discussed the difference between prokaryotic and eukaryotic cells. So this should be a review. Prokaryotic are smaller, simpler cells, no nucleus, and no other membrane-bound organelles, but they do have ribosomes. and that includes all the organisms in the domains bacteria and archaea. So these are all one-celled, small, simple organisms. Here's a diagram of just a generalized diagram of a prokaryotic cell showing the features that some prokaryotic cells have, but most of them don't have all of these things. But like all cells, it has a plasma membrane here, but it also has surrounding that a cell wall, and then to the outside of that is yet another layer called a capsule for protection, and in some cases the capsule is sticky and helps the cell, the one cell organism attach to surfaces or to other cells, other bacteria. This would be the chromosome, and in bacteria and archaeans, it's typically just one loop of DNA instead of the straight chromosomes like we have in our cells. There are often other little loops of DNA called plasmids that in many bacteria, that's usually just one gene and a little loop of DNA. So the little dots are the ribosomes where proteins are made. These short little hair-like extensions are called pili, pilus with the US is singular. And these are also for attaching to surfaces or to other cells. And some, when we do a chapter on bacteria, we'll see that there are some of these are hollow. It could be used by this cell to connect to another cell, and then they can one cell can send DNA into another cell through that sort of conjugation pilus. And then the flagellum or the tail. This is usually for swimming, and most bacteria and archaeans don't have that, but a few of them, a few species have that. And compared to eukaryotic cells, which are larger, more complex, have a nucleus and other organelles, and so these eukaryotic cells make up animals, plants, fungi, and protists. So that's what our human cells are, eukaryotic. Again, comparing the size, the bacteria cell is much, much smaller than typical eukaryotic cells. The cell size is limited, so why do I have to be made up of trillions of cells? Why can't I just be one big cell? And the usual accepted answer to that is that a cell has to have a lot of stuff coming into and out of it all the time, and so it has to have enough surface area to bring stuff in and to have stuff go out. And the larger an object like a round, this is a cell. Think of a golf ball, something the size of a golf ball, and then compare that to a basketball. The basketball is much larger. The ratio of the surface area to the volume, or the space inside a round object like this, this ratio is larger in a small object than in a large object. So in other words, the larger a ball gets, the less surface area there is compared to the space inside it, or the mass of living material in if this was a cell. So if the cell was this large, it just wouldn't have enough surface area for stuff to come in and out to supply all the living material inside. And so instead, large organisms like us are made up of many small cells. Okay, this is just showing the relative sizes of various objects, and this is not the scale. So this is a plant cell, and this is a human cell. This is actually much smaller compared. But on the scale here, it tells us this would be meters, millimeters, which are 1,000th of a meter, and then microns are also called micrometers, that are 1/1,000,000th of a meter, and then nanometers are 1/1000th of a meter. And here we see plant and animal cells about the same size. The human egg is a very large cell. It's about the size of a period at the end of a sentence in a textbook or something. And your mitochondria, this is one of the organelles that we'll talk about in this chapter that's inside of these cells. So that's what these little things are, mitochondria, I guess. The green ones are chloroplasts. So these are small parts of a eukaryotic cell, but they're but they're actually larger than a whole bacteria cell. That tells us how tiny these bacteria are. And this tells us here that you can see with a naked eye down to about 100 microns, and then light microscope is good for 1 millimeter down to 100 nanometers, and then an electron microscope, which is much higher magnification, is better for smaller things. Even molecules can be seen with an electron microscope. But looking at atoms and molecules with an electron microscope is kind of tricky because this device is shooting up beams of electrons at the object that you're looking at, and when electrons hit an atom, it's going to move the atom or disturb it or change it. So it's hard to look at something while you are moving it or disturbing it. So don't always get good images of atoms and molecules with an electron microscope. Okay, so here's an animal and plant cell compared. So animal and plant, both surrounded by a plasma membrane, but what the plant cell has that the animal doesn't have is the cell wall to the outside of the plasma membrane, and that's made of cellulose, polysaccharide. That's what plant fiber is. It's the cell walls of the plant cell. And all the nucleus, usually the largest organelle, and you can usually see that under a microscope as a dark spot. And other organelles, this is called the endoplasmic reticulum. It's kind of surrounding the nucleus. And Golgi, mitochondria, chloroplast. That's only in plant cells and algae. Um, uh, oh, this big sack of water here is called the central vacuole. Um, a vacuole. I'm looking for the word vesicle. I don't see it. A vesicle and a vacuole really mean the same thing. If the vesicle is small and the vacuole is large, but it's just a sac made of membrane. Same kind of membrane that the plasma membrane is made out of. But in the plant cell, the central vacuole has water, and often can be some other molecules stored in that water. It can be proteins or sugars stored in it, but it has an important function in plants that don't have wood. Plants that have wood, we call them woody plants, and the wood makes them stiff and helps them stand up straight. But many plants don't have wood. Soft stem plants, we call them herbaceous plants, but what helps them stand up straight is there's the water pressure in these vacuums. And that's why if you have a house plant and you don't water it, then it'll start to wilt because all these central vacuoles in the plant cells are losing their water pressure. If the plant's not dead, you could water it and it might stand back up. Okay, so we'll be looking at some of these organelles and see what they do. Plasma membrane is made up of phospholipids, bilayers. There's 2 layers of phospholipids, and has protein molecules and cholesterol also mixed in with the phospholipids. So cells always have to have a lot of stuff coming in and out. They have cells of your body have to have oxygen coming in, nutrients like sugars coming in, and waste products going out, carbon dioxide and other stuff. And there has to be the right amount of ions in the cells, like sodium and potassium and calcium. And if there's not enough, it'll have to have ions coming in. If there's too much of something, it'll be going out. So that plasma membrane is important for moving stuff in or out of the cell. So it's mentioned here that some cells have microvilli. These are only in a few places in a human body, but we have them in our small intestine. So if we take a cross section of your small intestine, which is a tube with food being digested, and it's made up of cells. So this food is being digested into molecules that are then absorbed through the wall of the intestine into blood vessels. These microvilli, they are little finger-like projections like this on this surface of these cells. And that's just for increasing the surface area for absorbing nutrients from the digested food. What else? We'll have a lot more to say about the plasma membrane, but here's a diagram of it. So this phospholipid bilayer is shown here. These little symbols here, each one of these represents a phospholipid molecule. Remember, that's similar to a fat or triglyceride molecule. It has a glycerol head, but instead of 3 fatty acid tails like a fat molecule, it has only 2. And the they are arranged in these two layers this way because, with the tails facing each other, because these phospholipid molecules have the head part is polar, like water, so it's hydrophilic. It likes to mix with water, likes to be in contact with water. And the tails are nonpolar, hydrophobic, so they don't like to be in contact with water. So when they arrange themselves with the tails toward each other and the heads in contact with the water, because the inside of the cell is watery, and the outside of the cell, uh, like the cell of your body, is surrounded by tissue fluid, watery tissue fluid. Um, and there's also cholesterol molecules. That's what these are. There's cholesterol molecules mixed in with the phospholipids. That helps make this membrane flexible. And then there are all these protein molecules embedded in the bilayer. And there are many different kinds of these proteins for different functions. Some of them are receptors for hormones. So if this is a cell of an organ in your body that is affected by a certain hormone, then it would have receptors for that hormone. Like with this. So if the hormone molecule would come along, like adrenaline would come along and contact that receptor, it would cause some kind of change in that cell to happen. Some of these proteins are like they're like ID badges on the cell, so that if this was a cell of your body, these identification markers would tell your immune system that cell is part of your body, and so your immune system should leave it alone. It's not a foreign bacterium or something disease-causing. But these ones that penetrate all the way through, this is a cross-section or cutaway view of this protein molecule, and it looks like it has a tunnel or a channel through it. Here's another one. So these are called channel proteins. They have tunnels or channels through them, and that's because some substances can easily move in and out through the phospholipid bilayer, like oxygen can pass right through that pretty easily. Carbon dioxide can pass through, water can pass through, but other stuff, like most ions, like sodium and potassium and calcium ions, they can't get through this, but they could get through these tunnels. That's how they come in and out. Or sugar molecules that we have to have coming into our cells for energy, they can't get through this phospholipid bilayer, but they can come through these channel proteins. Okay, the cytoplasm, the inside of the cell. So that's where the organelles are, and the organelles are they're all floating around in a jelly-like substance called cytosol. And the cell has something like its own skeleton to hold its shape and even to change shape. We'll talk about that a little bit in a minute. Mostly water in its cell, and always a lot of molecules in the cytosol, sugars for energy, and cells are always making molecules, proteins and carbohydrates and lipids. So there's all this stuff, polysaccharides, amino acids, for raw materials for making molecules. So these nucleic acids, like RNA, will learn about that have the information or that tell the cell how to make different molecules. A lot of chemical reactions going on inside of cells all the time. Okay, cytoskeleton. Let's just look at this picture, this diagram. So this is just showing a section of a cell. So this would be the plasma membrane on top and on the bottom. And this is the inside of the cell. And these long things are just protein filaments. And they make up the cytoskeleton. And there's three kinds. There's real thin ones called microfilaments, and then thicker ones called intermediate filaments, and then hollow ones called microtubules. But they're attached to the plasma membrane, and they're attached to organelles, and they hold the organelles in place, and they can get longer and shorter to move the organelles around. And in some cells that can change shape, they change shape by the cytoskeletal filaments getting longer and shorter. Flagellum and cilia. You have pictures? Pictures. So flagellum again, that's a tail, the long tail that some cells have, and it's usually for swimming through a liquid environment. And there's only one kind of human cell that has a flagellum, and that's the sperm cell. And they usually move like a corkscrew, and that's how the cell swims. In cells that have flagella, usually there's only one, or some kinds of one-celled organisms have two flagella. But if a cell has a lot of smaller structures like flagella, they're called cilia. Some cells, like a paramecium that you probably learned about. Paramecium is the one that's described as slipper-shaped. Something like this, and they're covered with cilia. And they are in a group of one-celled organisms called ciliates, because they're covered with cilia, and that's how they swim. These cilia always they move like they're doing a wave, all moving together for swimming. And in your body, you have some cells that have cilia, but not for swimming. It's for moving liquids or moving substances through a tube. They look similar to the microvilli that I sketched that we have in our small intestine, but the cilia are movable. And we have them in our a few tubes in our body. We have them in our airway here, our trachea that we breathe through. And those cilia are they project into the hollow airway that the air moves through, and they're covered with sticky mucus. And the air that we inhale from the atmosphere, it's got a lot of stuff in it. It's got a lot of dust and pollen and bacteria and mold spores, and just airborne. A lot of airborne stuff, and we don't want that going all the way into our lungs. So that most of it gets filtered. Big stuff like large dust and insects would get filtered by nose hairs, but then in our airways it gets stuck in that mucus on the cilia, and the cilia are always moving that mucus up. The cilia are always sweeping the mucus up our throats, and your airway that you breathe through, at about the level of the Adam's apple here, the voice box, the trachea that we breathe through is connected to the esophagus behind it that we swallow through. And so that we frequently swallow that mucus down into our stomach along with all the dust and stuff that that is stuck in that mucus. Um, Tobacco smoke paralyzes the cilia, so smokers, their cilia don't move; they just lie there, and so the mucus just, instead of the mucus being swept up um, the through the airways um, uh, to prevent it from accumulating down in the lungs, the mucus just slides on down into their lungs. That's why smokers cough a lot, and sometimes when they laugh, you can hear the mucus in their lungs because the cilia are not functioning the way they're supposed to. There's also cilia in the uterine tube, the fallopian tubes that move the egg from the ovary to toward the uterus after ovulation. They help to sweep it through that tube. I run every other day. I run. I've been running almost my whole life, and I last few years I've done most of my running at night. I don't know why I got in that habit. It's just more convenient for me. So I wanted to run with a headlamp, and about a year ago I got a new headlamp. It was really bright, very powerful, 800 lumens, and when I first time I used that thing, it's the light directed right down in front of my eyes, and sometimes I'm just amazed at how much stuff is in the air, because every little tiny particle is illuminated by the headlamp, and it looks like it's snowing sometimes. Just on a clear night, there's just so much stuff in the air, and I'm thinking, God, I'm breathing all that stuff. I should be wearing a mask just to go outside. But it's a good thing we have this system in our respiratory system to filter out most of that stuff. Okay, so the so-called endomembrane system, it's just a few different organelles that work together inside of cells to make molecules, and then deliver those molecules to where they're needed in the cell, or even to release them to outside of the cell. And so it involves these organelles that we'll talk about a little bit. So first, the nucleus. That's where the chromosomes live. So the chromosome says directs the synthesis of ribosomes and proteins. So cells are always making protein molecules, and then the protein molecules do most of the work in the cells, especially enzymes. And we learned that enzymes are special kinds of protein molecules that cause chemical reactions to happen in a cell. So the information for making a protein molecule is stored in the genes, in the chromosomes. So remember, protein is made up of a long chain of amino acids. The information in the genes tells the ribosomes, which make the proteins, which amino acids to put together in what order to make that protein molecule. And the ribosomes are made in the nucleus. Inside the nucleus is a structure called the nucleolus. So here's the nucleus, and this dark spot within the nucleus is called the nucleolus. That's where ribosomes are made. But the ribosomes are out in the cytoplasm, so there are holes in this is called the nuclear envelope, the double membrane surrounding the nucleus, and there are holes in it called nuclear pores So that would be how the ribosomes that are made in here would get out into the cytoplasm. So this would all be all chromosomes. These are very, very long. I mean, for molecules, they're really long. Some of these are an inch or two long. And think of that, almost every cell in your body has 23 pairs of these long molecules all folded up in there. What else? This stuff surrounding it says endoplasmic reticulum. We'll talk more about that in a minute, but this is another organelle. And these little dots are ribosomes that are attached to the endoplasmic reticulum. So the information for making proteins is here. The ribosomes are out here. There's also a lot more ribosomes that are just out free in the cytoplasm. They're not all attached to the endoplasmic reticulum. So vesicles and vacuoles, already talked about that. These are just little sacs within a cell for containing stuff or moving stuff around, and it's just made out of membrane material. Same phospholipid bilayer as the plasma membrane. Please wake up. You don't get credit for attendance if you're asleep. The endoplasmic reticulum, just call it the ER for short. So it says interconnected membranous tubules. So here's what it looks like again. This is all membrane material, and it's in the form of these flattened tubule tubes or sacs. And there's two kinds. The rough ER has the ribosomes attached, and it's called rough because it kind of looks like sandpaper under a microscope. The smooth ER does not have ribosomes, and ribosomes are for making proteins. So the rough ER makes proteins. So these ribosomes, they make proteins, and then the protein molecules end up inside these flattened sacs or tubes. Other kinds of molecules like lipids and carbohydrates are made in the smooth ER. Then these molecules that are made by the ER are then put into vesicles, little sacs made of membrane. And so part of this membrane of the ER will just pinch off and form a vesicle with the new molecules inside of it. Now these new molecules, they might not quite be finished being made, and so they next go into this next organelle called the Golgi apparatus. Some books call it the Golgi complex. The Golgi, I guess, named after a guy named Golgi who discovered it or something. But so this vesicle is called a transport vesicle because it's for transporting or moving this new molecule. It goes to the Golgi. Now the Golgi will finish those, making those molecules. For example, we learned that every protein molecule is made up of chains of amino acids. So maybe the chains of amino acids are made here in the ER, but then they are put together in the Golgi to finish that protein molecule. Now the Golgi will repackage them into vesicles. Now these are ready to go wherever they need to be used in the cell or outside of the cell. In this case, this vesicle is going to the plasma membrane, and so this membrane will just fuse with this plasma membrane and just release, just spits the stuff out to the outside. So that would be That would be happening in cells of glands in your body. A gland is an organ that produces something. It could be a hormone or saliva or earwax or tears. So the cells in those glands are making that stuff, the hormone or saliva, in the cell, but then it releases it to the outside of the cell, into the body, the person's body. Um, what else about the ER? The smooth ER, like I said, it's mostly for making molecules, but in some cells, some human cells, um, uh, uh, like cells of the liver, it's the smooth ER functions in breaking down toxins and drugs. So many of the kinds of drugs we take, um, they don't. You take drugs, they don't stay in your body forever. What happens to them? Your liver sees them as toxins and so breaks them down. Um, Alcohol, when you get drunk, you don't stay drunk forever because your liver cells um, break down the alcohol. How long does it take to get drunk? How long until that alcohol is cleared from your body? Do you know? About 10 hours. Depends on how much, but 5 to 10 hours. If you get really drunk, 10 hours. Um, Alcohol is a toxin. Uh, in the muscle cells, there's a special kind of smooth ER that stores calcium ions, and for a part of the process of a muscle being triggered to contract involves calcium being released into the muscle cells from the smooth ER in those cells. Okay, so ER and then Golgi talked about that. Under a microscope, the the ER and the Golgi look the same. They're just these flattened tubules like this. I can't tell one from the other, except that the ER is usually up against closer to the nucleus than the Golgi. So all this stuff together is called the endomembrane system. The nucleus has the information for making molecules. The molecules are made here in the ER, and then they're finished in the Golgi, and then they're put into vesicles to go wherever they are needed. Okay, it's a micrograph of the Golgi. Okay, other organelles, lysosomes, the garbage disposals of the cell. That's what you need to remember. So this is where unneeded molecules are broken down, or organelles that the cell doesn't need anymore. They'll be broken down, broken apart in the lysosomes. So these are like vesicles containing enzymes for breaking molecules apart. Mentioned here that this phagocytosis will define that later in this chapter, but phagocytosis means a cell eating something. Some cells are capable of we have a picture here. Capable of doing this. The cell can change shape and wrap around a particle that's outside the cell and take it in. Not all cells can do this, but some types of white blood cells of your immune system can do this. And so if they find a bacterium or something that doesn't belong in your body, they'll gobble it up like that, and then put it into a lysosome to destroy it, break it down. And so this is called phagocytosis. The large white blood cells that can do this are called macrophages. Okay, then ribosomes. This is where the protein molecules are made. And I think in another chapter we'll we'll see how the ribosomes do it. How do they use information from the chromosomes to put amino acids together to make proteins. An example here of a protein that's made in some cells is hemoglobin. This is in our red blood cells. In fact, this is why your red blood cells look red to us, because hemoglobin contains iron and it makes it look red. But we inhale oxygen into our lungs, and that oxygen goes into our blood, and then the oxygen molecules attach or hitchhike on the hemoglobin while they're in the blood. Hey, mitochondria. We're going to do a whole chapter on this. I think chapter 4 or 5. On this process called aerobic cellular respiration that occurs in the mitochondria. And this is a chemical reaction for breaking down nutrient molecules like sugar to get the energy out of them to keep the cells alive. And those chemical reactions require oxygen. That's what aerobic means. Requires oxygen. And so these are called the power plants of the cell because it's kind of like burning coal, like a coal burning power plant. The fuel is coal and it's burned to produce heat energy. And burning is, we said before, burning is a chemical reaction between some kind of fuel and oxygen, like coal and power plant. So it's kind of the same thing that happens in mitochondria. It's like burning sugar using oxygen. Except it's not to produce heat. It's a very, very strictly controlled kind of chemical reaction, and the energy is then stored in ATP to be used by the cell. Remember, these are like little rechargeable batteries that are always being recharged in the mitochondria and then used for energy elsewhere in the cell. You tell this here. Cells that use a lot of energy, like your muscle cells, it takes a lot of energy to do this, to move your body parts. So they have lots of mitochondria. Brain cells have a lot of mitochondria. So they look, the mitochondria are kind of long oval shaped or shaped like sausages or something. Peroxisomes, so just think of these as similar to the lysosomes, like garbage disposals. Again, they're for breaking down cells that are not needed or even toxic molecules in the cell. And they're called peroxisomes because they produce hydrogen peroxide as they are breaking down. And there's this, in fact, something I could have mentioned, but for lysosomes, where's that name lysosome? The word, the Latin root lys means to break apart or to break down. So lysosomes. So if you ever see something like lysis in a word, it has something to do with breaking something down. Okay, comparing plant and animal cells. Again, there are some, they have, both plant and animal cells share a lot of the same organelles. They both have mitochondria and endoplasmic reticulum and Golgi, but lysosomes are only in animal cells, not plants. These centrioles and centrosomes, we didn't mention those. These are part of the cytoskeleton. And they only function when a cell is dividing. And so we'll mention them when we learn about cell division, mitosis and meiosis type cell division. So when a cell divides, the chromosomes have to move into the separate daughter cells And that's what these do. They they make the cytoskeleton fibers that attach to the chromosomes and then move them into the daughter cells as the cell is dividing. Um, Plant cells have a cell wall. We saw that earlier. Animal cells don't have a cell wall. Chloroplasts, those are little green organelles where the photosynthesis happens in plant and algae cells. Um, Plasmodesmata, these are connections between neighboring cells in plants that they're like tunnels connecting two cells so that stuff like sugars can move from one cell into the other through these tunnels. The plastids and vacuoles, those are like, again, sacs containing stuff that are stored in the plant. Okay, cell wall, it's made out of cellulose in plant cells. Other kinds of organisms have cell walls, like fungi have cell walls, but it's not made of cellulose, it's made of stuff called chitin. The same stuff that the exoskeleton or the skin of an insect is made out of this stuff. Bacteria and archaeans have a cell wall, but it's made out of this stuff called peptidoglycan. So the cell wall makes the plant cells more stiff and rigid. Okay, plastids again. The chloroplasts are considered to be plastids. Plastid being like a vesicle, it's a membrane containing stuff, but the chloroplasts are very special. That's where the photosynthesis happens. And we do a whole chapter on photosynthesis. The chloroplasts appear green because they have this green stuff called chlorophyll that absorbs the light, the sunlight, for photosynthesis. So both plant and algae cells have chloroplasts. There's a types of bacteria that are photosynthetic, and they're called cyanobacteria, but they are prokaryotic, so they don't have organelles, so they don't have chloroplasts. The photosynthesis occurs on the inside of the plasma membrane. It's not in chloroplasts in the cyanobacteria. These words autotroph and heterotroph. Auto means self. Hetero means different or other. So autotrophs make their own food. So that's what plants, photosynthetic organisms, they can make their own sugars and other organic compounds just using inorganic carbon dioxide and water. They can make sugars and then store that for later, use it for food. Heterotrophs, that's what we are. We have to eat other organisms, plants or other animals. So there's a chloroplast. We'll learn about them in another chapter. Okay, talked about this large central vacuole. It's like a water balloon inside of a plant cell, and that water is under pressure. The pressure is called turgor pressure. Just the pressure in a vacuole. We'll do a whole chapter on plants. And then this theory of endosymbiosis. Endo means within, symbiosis means some interaction between two organisms or two things. So this theory answers the question of where did the first eukaryotic cells come from. It's believed that life began on Earth 3.6 or 3.8 billion years ago, and they were prokaryotic cells, very simple bacteria type cells. But eukaryotic cells didn't appear until about 2.1 or 2.2 billion years ago. So for 1.5 billion years, all life on Earth was just prokaryotic cells, one-celled little simple organisms living in water. And then eukaryotic cells came along. How did that happen? Cells that had a nucleus and other organelles. And it's believed that the nucleus happened just by the plasma membrane somehow is folded in around the chromosomes. So here's a cell, and it had this chromosomes free in there. Somehow if this plasma membrane would fold in, would fold in a couple of places, I guess, and then completely surround the chromosomes and pinch off inside. Now that would be a nucleus, and that would explain why the nucleus has a double membrane, because of the way it would have folded in like that. But what about the other organelles? It's thought that chloroplasts and mitochondria started out as other free-living prokaryotic cells that got into they pushed their way into another cell, and so this cell would just push its way in until it was in here, and that would explain why chloroplasts and mitochondria have a double membrane surrounding them, because of the way they would push in and the plasma membrane would wrap around it. And these chloroplasts and mitochondria, so I think they were the first organelles, and there's other evidence. Chloroplasts and mitochondria, they still look like bacteria cells. They're about the same size. They have their own DNA. You may have heard of mitochondrial DNA if you watch forensic shows. But that's DNA that's separate and different from the DNA in your chromosomes of your nucleus of the cell. And when your cells make more mitochondria or chloroplasts, the ones that you have divide just like bacteria cells do. So they still look and act like separate cells. Okay. We're done. So exam next Friday, homework due Monday.